Hi everyone, welcome back to Useful Genetics. This is Lecture 1.0, where we're finally getting back to talking about genetic variation in populations. We'll talk about alleles and genotypes in individuals and populations, and we'll consider kinds of DNA sequence differences, because most of the analysis I'll be talking about is built on looking at sequence differences. We'll talk about how we compare DNA sequences, and we'll think about allele frequencies and genotype frequencies, specifically in populations, introducing some new terms and a new concept, the concept of a haplotype, which is a kind of genotype. Now, we'll start by considering a position somewhere in the genome in the DNA sequence. And I've shown this initially showing both strands with bars indicating the base pairing, but we don't need any of this because we know the conventions that when we see a single sequence of a single strand of DNA, that the left end is the 5' end, that the complementary strand would have the complementary sequence. We don't need to show any of this. Now let's think about genetic variation. How many different alleles are possible at this one position? Well, 4 alleles are possible because there are 4 bases. We could have an A or a G or a C or a T. What about in a person? How many different alleles could one person have? Well, a single person could only have 2 alleles because they only have 2 versions of the sequence. Now, how many different genotypes are possible for one person? If we're just considering they can only have 2 alleles, let's just consider the alleles A and G. In fact, there are 3 different genotypes possible for a person at any one position if we're just considering 2 alleles. And I'm now going to introduce 2 new terms which we'll be using quite a lot in the future. And so a person, considering 2 alleles, the person could have 2 copies of the identical allele. So they could have 2 A's, one from mom, one from dad, or 2 G's, in which case we would say they were homozygous at this position, this locus, either homozygous for A or homozygous for G. Or they could have 2 different alleles, in which case we would say that they were heterozygous at this position. Now let's branch out and think about the same position, but this time we're thinking about a population of people. How many different alleles are possible? Well, again, there's only 4 bases, so there's only 4 alleles possible. How many different alleles could the population have? When we had a person, they could only have 2, but the population could certainly have all 4 alleles. How many different genotypes would then be possible in the population? Well, with 4 alleles, we can make a lot of combinations. In fact, there's 10 different genotypes that would be possible just from the 4 different bases in a population. Now I've got 2 questions for you to answer. How many different alleles of a gene can a population have? And notice that, again, this is a question with square answer boxes. So you can check more than one answer if you think that's correct. And I hope you check more than one answer. The right answers are, well, it depends how big the gene is, and it depends how big the population is. Because a big gene, there are potentially 4 different alleles for every base in that gene. So that's many, many thousands. If the gene is 1,000 base pairs long, that's many thousands of alleles. If the population's very small, the population can only have 2 alleles per person. So the number of alleles can't be larger than twice the size of the population. So here's another question, just a different version. This is a classroom version. How many different versions of human chromosome 11 would there be in a class of 200 students? And I'm giving you information about how big the chromosome is in case you need it. And the answer is, oh, you didn't need to know how long the chromosome was at all. There's only 200 people in the class, there can only be 400 total versions of the chromosome. It's unlikely that any of them are going to be identical because the chromosome's so big. Every one of them is almost certainly going to be different. So 400 is the right answer. Now let's think about kinds of DNA sequence differences. We already introduced the concept of alleles, different versions or variants of a gene. But now we're going to think about alleles in the context of DNA sequences. And a term that we commonly use is a single nucleotide variant, often abbreviated as an SNV. That just means the presence in a population of different nucleotides at homologous positions in the two DNA sequences. So we have to be comparing homologous positions. We can tell that in this diagram here. First, we can tell that this diagram is a diagram of two different DNA sequences being compared. It's not a diagram showing the base pairing between two different strands of one DNA molecule. And we can tell that first because it's labeled genome 1, genome 2, not 5' end, 3' end. We can also tell very clearly because at any given position, the two bases are identical. There's a G here and a G here. There's a C here and a C here. At every position, except for one, the bases are identical. And at every position where the bases are identical, there's a vertical line. And that tells us that these vertical lines mean this is a position where the two sequences, the two genomes, have identical sequences. But there's one position where the genomes have different sequences. That's here. And that represents a single nucleotide variant. It's just one position where there is variation. Two different versions are present. Now, there's another kind of DNA sequence difference that we need to be aware of. And that's got the rather corny name of indel. And I'll explain in a second where that word comes from. So what we're talking about here are genetic differences not created by changing one base to a different base, but by inserting or deleting bases, one or more bases, into a DNA segment, which changes the length of the DNA segment. So you can see, again, we're comparing two DNA sequences, but one's longer than the other. The term indel is used because when we're comparing two DNA sequences and we find that they're different lengths, we don't know that the difference, which is marked here, arose because something was inserted into this sequence or because something was deleted from an ancestor of this sequence. And indel is just a compromise word in del because we don't know which it is. Now, when we compared two DNA sequences, I emphasized in the previous slide, they needed to be homologous. And that means, from our definition of homology from earlier, that they're similar because of shared descent from a common ancestor. So when we're comparing two positions, we want to be sure that those two positions are different, the same or different versions of a base, that they were both inherited from a single position in a common ancestor. We don't want to be comparing what were different positions in a common ancestor. So let's compare these two sequences. And I'm telling you that these sequences are homologous. So let's compare them. We draw a vertical line for each position where the sequences are identical and go, oh, they don't look very homologous. I mean, we expect one base in 4 to match just by chance. This doesn't look very promising at all. Maybe they're not lined up right. So let's shift them over one and try again. No, that's still pretty bad. Okay, let's shift them over a bit more. Aha! Now we have the right alignment. And we see that almost every position matches. Now we're confident that the homologous positions are aligned. And that's critical when we're comparing sequences. You won't normally have to do any alignments yourself, but you really should be aware that this is important. And the reason it matters is because some sequence variation is indels. Insertions and deletions are fairly common in chromosomes, and you can't just start at the end of two homologous chromosomes and just line up all the bases and compare them. You have to make sure in different positions that they're correctly aligned so the alignment reflects homology. Now, it's time to think about the frequency of alleles and genotypes in a population, because this will be critical for a lot of the analysis that we do. In particular, it'll be important for the last lecture when we talk about the evolution of human genetic differences. So the allele frequency in a population is really, really simple. It's just the number of occurrences of that allele divided by the number of total alleles in the population. That's the frequency. Sounds easy, but what if you don't actually know the frequency? You haven't counted the alleles. What if you've only counted the genotypes? What if you surveyed the population, you took a sample of people, and you determined their genotypes at a particular position that you're interested in, and these are the numbers that you got? Can you calculate the frequency of one or the other allele? Sure, it's easy. So we know the denominator. We know that there are 92 people. Each person has two alleles, so that's 184 alleles in the whole sample of the population. And we're assuming that this is a large enough sample that it's representative of the population. What's the frequency of the T allele? Well, 22 people, so 57 people don't have a T allele at all. We don't need to worry about them. 22 people have one T and one C allele. So that's 22 T's. And 13 people have two T's. Those people are going to contribute 26 T's. So 22, 26, 48 T alleles tells us that the frequency of T is 0.26, 0.26. So that's not hard, that's simple arithmetic. What about the other way around? What if we want to think about genotype frequencies in a population? Well, the basic calculation is still really simple. It's just the number of occurrences of a particular genotype out of the total number of genotypes, which is the total number of individuals in the population. But what if we only know the allele frequencies? Often when you're given a description of a population, you're given the allele frequencies, but not the genotype frequencies. Well, again, the calculation is pretty simple, but I'm going to show you a diagrammatic way to do it. So in this case, let's assume that we have the allele frequencies for two alleles in the population, and that the other two alleles aren't present. As you'll see, this is a typical case for real alleles. And we want to know the frequency of the AT genotype. Now, I'm going to show you a trick that might look kind of like the Punnett squares that you might have encountered if you learned basic genetics at some point, except I'm drawing the square off center. And it's just a way of illustrating what happens when alleles come together at random, as if when people married each other and had children, they did not check their genotype at this particular position in their genome before they did it, which is the normal case for almost every aspect of our genome. Mating is random with respect to genotype. Then we can just predict, just from basic probability, the frequencies of the genotypes that are going to be present in the population. So we know that 0.8 of the population has A's, and we're going to draw that A 0.8, and 0.2 of the population has T 0.2. And we can simply then look at the picture and say, oh, yeah, of course, the chance of A meeting A is going to be, to give the AA genotype, is going to be 0.8 squared, which is 0.64. And the chance of T meeting T to give the TT genotype is going to be 0.26 squared equals 0.04. Now what we're interested in, though, is the chance of T meeting A, or A meeting T. And these frequencies are, again, just the product of the frequencies of the alleles. So it's 0.8 times 0.2 equals 0.16 for this one and for this one. So now we've got two occurrences of this genotype. They're just written in different orders, but it's the same genotype. So we're going to multiply this number by 2 to get 0.32. That's the frequency of the AT genotype. And we can check easily that we've done the arithmetic right by checking whether our numbers add up. So we've got 0.4 of those and 0.64 of those and 0.32 of those. Does that add up to the whole population? Yes, it adds up to 1. So we can be confident that we've done the calculation right. Now that we know how to think about allele frequencies and populations, I have to introduce some terminology. If a genetic difference is present in less than 1% of the population, it's usually described as being a rare variant. But if it's present in at least 1% of the alleles in the population, there's another term that's very widely used. It's called a polymorphism. Until recently, geneticists have spent a lot more time studying polymorphisms than studying rare variants, just because they're a lot easier to find and to study. We're only now developing the tools to study rare variants, and we'll talk a little bit about this in Module 5. Module 5, yeah, I think that's right. So now if we introduce into this, again, the single nucleotide variants. Remember, single nucleotide variants are differences that are affecting a single position in a DNA sequence. Rare single nucleotide variants, we can have rare single nucleotide variants, but as I said, they're hard to study. The easy kind of variation to study, and by far the most common variation in the genome, are what are called single nucleotide polymorphisms, abbreviated SNP, and because they're talked about a lot, we have a way to say this. They're called SNPs. And SNPs are very common variation in populations. They're very easy to study. They're the foundation of all of the personal genomics that we'll introduce in Module 6. So this is a very important concept to get straight. If these two variants are, if each of these variants is present at least 1% of the population, this would be a SNP. If this was very, very common and this was very, very rare, we'd say, well, it's a rare single nucleotide variant. We're not going to study it. It's too difficult. Now, most polymorphisms have only two common alleles, like the polymorphism that I introduced when we were doing our arithmetic. Here's an example. This is a small segment of the BRCA2 gene that we described earlier that includes high-risk alleles that cause a high risk of breast cancer. And this is a diagram showing the known SNPs, the known single nucleotide polymorphisms, in this segment of the genome. That's about 20 kilobases long. And I'll circle the variations so you can recognize the diagram. It's a little tricky to interpret. Each vertical pair describes a SNP at that particular position. So here there are 3 different SNPs present at this position in the intron. Here are 3 SNPs in this small exon. Here are 5 SNPs in this exon. Here's 1 SNP in the middle of the intron. And you can see that there are lots of SNPs, both in introns and in exons, and that there are always only 2 alleles are common. There may be rare individuals who might have a C or a G, but they're sufficiently rare that they don't count as polymorphisms, and they're not normally considered. Now, I want to introduce one more concept, and that's the concept of a haplotype. We need this for the next lecture. So a haplotype is just the genotype. Instead of being the genotype of a particular position, it's the genotype of a segment of the chromosome. It's used in slightly other variations in other contexts, but for us, for now, this will capture it nicely. So here we're showing the sequences of a little bit of the chromosome of the genomes of two different individuals. And we see that, oh, there are 4 different places where they have different bases in their DNA. We would describe this collection of variation, this segment of variation, as a haplotype. So we could say this is the haplotype that has a T at this position and an A at this position, sorry, a T at position 2 and an A at position 16 and a T at position 20 and a C at position 28. This is a haplotype that has a G at position 2 and a G at position 16 and an A at position 20 and a G at position 28. So this has been a long lecture, the longest lecture of Module 1, and we've introduced a lot of new material building on the previous lectures. We talked about kinds of sequence variation, how we can compare DNA sequences, why it matters how we line up the bases, because some sequence variation is changing the lengths of DNA sequences. We talked quite a lot about alleles and genotypes in populations. We did some calculations, and we introduced a number of new terms: homozygous and heterozygous, polymorphism, SNP, indel, haplotype. These terms are going to come up again and again in the course. Now we're ready for the last lecture of Module 1. We're coming back to the question of how genetically different are different people, and we're going to use that and some advanced DNA sequencing techniques to answer the compelling question of whether or not our ancestors really did have sex with Neanderthals. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is the last lecture of Module 1, where we're coming full circle back to thinking about human genetic differences. And we'll be using these differences to get a deeper and an evolutionary picture of the genetic history of our species. Now, we'll start by basically where we left off in the first lecture, thinking about how different are different people. And we said at that time that the average 2 people on average are almost identical, that their DNA differences are only about 0.1% of all the sequence positions in their genome. In this, we're actually a little more similar than most plants and animal species, where on average there's about 99%, not 99.9% sequence identity. We can see this is because we're actually quite a young species in the average ages of the species that are alive today. We arose relatively recently, and we haven't accumulated as much difference. And we can see the whole range of within species differences in this rather overwhelming chart. Basically, this is the extent of sequence difference from about more than 5% in some worms down to very, very little genetic difference in some species that have been recently very highly inbred. And we'll talk about this inbreeding a lot later in the course. But here's us. We're definitely down at the low variation end of the spectrum of species, which is thought to reflect our recent origins as a species. Now, insights into our origins as a species can come from thinking in a larger way about the genetic differences between different people from different parts of the world. And I want to describe an important study by Dr. Sarah Tishkoff and her colleagues. And what they did was they analyzed not just single base differences, but haplotypes. And remember, haplotypes are segments of DNA with genetic differences. So all the genetic differences in a segment. In this study, they looked at their segments were 4 different genes, and they sequenced these 4 genes in 1,400 people from different places around the world. And they counted up the number of different haplotypes that they found in those 1,400 people. And they found 98 different haplotypes in Europeans, 73 in Asians, and 199 in Africans. So already we can see that, well, there's more genetic variation in Africans than there is in people from other parts of the world. This is actually consistent with other studies of human genetic differences. But more insight came when they thought about how much of these sequence differences were actually shared between the different groups. And what they discovered was that most of the genetic variation that they found in Europeans or in Asians was also present in Africans. And this suggested that Africans were the older and more diverse peoples, and that Europeans and Asians represented a subset of the genetic diversity in Africans. So this result was consistent with and strongly supported a view of human evolution called the out of Africa model. And that model proposed, and we are quite confident that this is indeed true, that that humans originated in Africa, and that maybe 150,000 years ago, humans spread through Africa, and maybe 100,000 years ago, a particular group of these people moved out of Africa into southwestern Asia, what we call the Near East. And then they gradually spread over thousands and thousands of years into Europe, into southern Asia, into northern Asia, into Southeast Asia, and to Australia and the Pacific Islands, and up through Eastern Asia, across the Bering Land Bridge, and down into North and South America. This is strongly supported by many lines of evidence. But what I want to tell you about now is new evidence that adds a new wrinkle to this out of Africa model. And that's addressing the question of, well, did our ancestors really meet with Neanderthals? You remember that Neanderthals are these extinct type of humans with big jaws and heavy brows. They're the canonical sort of caveman morphology. So thanks to a wonderful body of work by Professor Svante Pääbo of the Max Planck Institute for Evolutionary Anthropology, we know that the answer to this question is yes. And he describes his work very nicely in a TED Talk that's on the Module 1 webpage. So here's my drawing of a summary of what we know about what happened in human evolutionary history. Again, early humans originated in Africa, and within Africa, they spread and diversified. And at some point, a subgroup of these people migrated out of Africa over, this was several 100,000 years ago. These people migrated out of Africa into Europe and Asia. And these are the people that we call the Neanderthals. At the same time that the Neanderthals were colonizing this new land, evolution was continuing in Africa. And in particular, the early humans were gradually replaced by what we call anatomically modern humans, people whose facial features, whose skull structures are much more like ours. And again, the same thing happened as happened before. The anatomically modern people gradually, one branch of them, they spread through Africa. But one branch of them migrated out of Africa, again into Southwest Asia in the Middle East. And we now know that at this time, they encountered the Neanderthals. So the early humans in Africa were gone, but the Neanderthal people in southern Europe and Asia were still there. And so they encountered them, they mated with them, they had children. Their children became part of the wave of anatomically modern humans that was spreading into Europe and through Asia, into Northern Asia, into Southeast Asia. And again, in Southeast Asia, the anatomically modern humans that were migrating, moving across Asia, again encountered Neanderthals, and again they mated with them, had children, and their children became part of the population that was extending into Southeast Asia. So the anatomically modern humans continued to spread, including spreading over into North and South America to be the native peoples of those continents. But the Neanderthals, like the early African humans, died out. So we know that the ancestors of non-African humans mated with Neanderthal humans before the Neanderthals went extinct, and that some Neanderthal haplotypes live on in the non-African genomes. So African peoples don't have Neanderthal DNA. In a sense, we could say their genomes are more modern than ours. Our genomes, ours being people like me, of course, have Neanderthal DNA in them. Now, this is pretty detailed considering none of us were there to watch this happen. How do we know this? Well, we know this because Dr. Pablo's team has developed sophisticated ways to sequence DNA from ancient bones, even though that DNA is very badly degraded. It's broken, it's damaged in many ways, but they can sequence it. And they've now taken Neanderthal bones from six different sites, I think more now, including one from a site where the peoples who are related to the Southeast Asian interbreeding happened. And they can identify Neanderthal haplotypes by comparing their Neanderthal genomes with human genomes. They can identify all of the Neanderthal haplotypes across the genome. And what they find is that the genomes of non-African people contain 2 to 3% Neanderthal alleles and haplotypes. Not always in the same places, so different ones, we often have different pieces. And the genomes of the natives of Papua New Guinea and Australia, that's the South Pacific natives whose ancestors participated in the second interbreeding, their genomes contain about 6% sequence patterns from the Neanderthals that they mated with. So what we've done, we've talked about human similarity and human differences, compared ourselves to other species, and then we've done a more detailed analysis of human genetic differences, looking at haplotypes, not just at single position differences. And analysis of these haplotypes in modern people confirms the out-of-Africa model for the evolutionary history of humans. And with Dr. Pablo's techniques for sequencing ancient DNA, also confirms that some of our ancestors mated with Neanderthals in their migration out of Africa. Of course, those of us who are Africans, you're not involved in that. Now, again, I want to strongly recommend Dr. Pablo's TED Talk. It's 20 minutes long. It summarizes in its introduction just about everything that we've talked about in this module. And he's a wonderful dynamic speaker, so you'll find it well worth your while as a refresher for the whole module. Now, coming up next is Module 2, where we're going to talk about how DNA sequences change, the mutational processes that generate all this diversity. I hope to see you there.